So here we are ready to start chapter 6. First we'll talk about energy, we'll talk about the energy carrier, ATP, and then we will, the next screencast, cover enzymes. So here we go. We will be talking about catabolic and anabolic pathways eventually. Your book throws those terms in now, so I think we can introduce them in terms of energy and how we use energy. I like to talk about them in the sense of digestion. So catabolic pathways are when we break apart our food. We eat some sugar, we break it down into the monosaccharides, we release some energy, and then we further break down the sugar, and we'll hear about that in the next chapter. That's a catabolic pathway, breaking things down. You can think about it as this side of these pictures where we are using energy. You can release energy to do work. We'll talk about that more. Anabolic is building up or making order, using energy to make something, to create this beautiful order in our world, to make molecules, maybe make the polymers, make the proteins that need to do work for you. And I always think of anabolic bulking up. You always hear about anabolic steroids. They're the ones that are taken inappropriately to bulk up muscle. So let's talk about some ideas on energy. From your physics classes, chemistry classes, you should be aware of these words. This person has potential energy. When they dive, they convert their potential energy into kinetic energy, and that kinetic energy is driving the muscles. And then this person down here has less potential energy when they hit the water. But we're going to talk about a lot more in biochemistry is chemical energy. You'll see some examples of that. And the book just talks about, and we're not going to get into it in really gory detail, the first law of thermodynamics, that energy is neither created or destroyed. It's always conserved. And then the second law of thermodynamics is that when we do release that energy, some is dissipated as disorder and heat, and you'll see the energy equation in a minute. So when this bear breaks down sugars and glycolysis in the electron transport chain, cell respiration, some of the energy is released as heat, and the bonds are broken to release the CO2 and the H2O. Then that's just the order that we can create when we have extra energy to do some work. So forgive me, I don't have the delta sign easy on this software that I was using. So this is the Gibbs free energy equation. So delta G, so change in G, is equal to the change in heat, or enthalpy, minus the temperature times the change in entropy. And entropy is a measure of disorder. Basically, if you look at this glucose molecule and you release its chemical energy, you get more disorder in the system. Things spread out, things are less organized. You need energy to go the other way. Same thing, diffusion. Think about somebody showing up at one end of the room with some perfume with molecules in a very concentrated area. They diffuse and they become more disordered and release some energy as those molecules bounce around. So there is lower free energy here. G is smaller. G is higher. Gibbs free energy. And we need the free energy to do work. So let's get into what that work is. So you might remember these types of diagrams from chemistry, I hope. This would be progress of the reaction, so how the reactants turn into products. In this case, it's called an exergonic reaction. It's releasing energy. We are starting out at a higher G, we are ending up at a lower G, so the change in G, again I should have the delta sign here, is less than 0 or a negative G. It means that energy was released from the system. This is also called the spontaneous reaction because you release energy. The opposite of this would be an endergonic reaction. The reactants start at this lower level. Energy is put into the system, more order is created, and the products are up at a higher G. So this delta G is greater than 0. Energy is required in the system. These are gonic for G, endergonic. This is not thermic, endothermic, which you might have heard about in chemistry. Kind of feels the same, but it's actually a slightly different reaction. So endergonic is you need energy to build things up. So we could talk about different kinds of systems. If you had an isolated, they use an example of this hydroelectric system where the turbine's going to run as the water level goes down, make some energy. In this case, the delta G is less than 0 because we're making energy. And eventually, because this is a closed system, it's going to hit equilibrium. It turns out that in living systems, if we are ever at a delta G equal to 0 or equilibrium, we are dead. So open biological systems are actually open systems. There's always something going in and something coming out. Very simple one here, that you're always putting water in, you're always taking some water out, so delta G is less than 0 and you're always doing some work. This one, this multi-step, again, this is just a cartoon to get a point across. You're always putting something in, you're always taking something out of this. There's the multi-stepped one. This is a lot more like a human body or a cell where there are many different things going on, releasing energy to have it have that whole processes taking place. So what do we use to move energy around in the system of the cells? The currency of the cells, so to speak, is called ATP, adenosine triphosphate. So you should recognize quite a few of the functional groups. Basically, it's an adenine nitrogenous base, and that's hooked to a ribose. So we recognize this a lot. There's a sugar. It has the 2 oxygens, so it's a ribose. And then on that 5th carbon, we have 3 phosphates attached. Each phosphate has a ton of energy in this bond holding them together, predominantly because these negative charges are not very stable in that covalent. That covalent bond is stable, but it really wants to fall apart. And when it falls apart, it actually releases quite a bit of energy. About 7.3 kilocalories per mole of energy gets released. So this is the way your body and your cells move energy around. This evolved very early in life, and we use it from bacteria all the way up through humans. We'll be talking a lot about how ATP gets made in the next chapter when we talk about glycolysis, the Krebs cycle, and the electron transport chain, or cellular respiration. So again, if this is our ATP molecule, now it's been abbreviated a little bit, color-coded. If water comes in, usually an enzyme is going to be involved with this case. Phosphate can get transferred. When you break that bond, you release this glowing orb of energy, and you're left with ADP, or adenosine diphosphate. And these guys are going to cycle back and forth. There'll be a process to put that phosphate back on that costs energy. You release it again, you release energy. Now here's a nice example of how you can use the energy from ATP in a coupled reaction to make something happen. So for example, we can build a few amino acids in our body. We eat quite a few. We recycle them as we digest them, and then sometimes we can make some fresh ones when we need them. So glutamic acid, as this triangle, can be attached to an ammonia molecule to make glutamine. This costs the cell, or the reaction costs 3.4 kcals per mole. So how do you accomplish that? You basically couple the reaction with the hydrolysis, or the ripping apart of an ATP. So first, it's a multi-step thing where we're transferring that phosphate and making something unstable. So the glutamic acid grabs the ATP and grabs one of those phosphates. So energy gets released in this situation, and then the ammonia group displaces that phosphate. It's basically more reactive, and we then get our final product. So again, we're going to see how we can offset that 3.4 kcals per mole. And it turns out that by ripping apart your ATP, this part of the reaction, you release 7.3 kcals per mole. So if you go minus 7.3 kcals per mole plus 3.4, we're still in the negative. So that's a spontaneous reaction that takes place. This is just an example of how ATP can be used in reactions. You've already heard in the membrane, why that always does it drives me crazy. We've already heard in the membrane section that ATP can be used to phosphorylate a transporter protein and force molecules against their gradient. Remember, this is active transport. So here's one very important use of ATP. And another use is to power these motor proteins. We have microtubules in the cell you just heard about recently, and we have vesicles that get carried along those microtubules. And here's a little video of how the microtubules on the bottom, I'm going to try to keep my cursor up here, microtubules are on the bottom. There's the dynein molecules that bind ATP and then release it as ADP plus phosphate. So now here comes a new ATP in. It binds that one. It changes the shape of the molecule and makes it swing around to grab onto the tubulin molecules. And then we release the ADP and phosphate, and we do it all over again. And basically now we can have this motor protein literally walking along the microtubule. So each time it takes a step, it costs an ATP. That's a pretty expensive process. ATP can get regenerated in the cell. So again, we can use up our ATP, we can release some energy, and then we can recycle that ADP plus phosphates floating around, diffusing in the cell. And we're going to see the mechanism that gets rehooked up, a couple different ones in cell respiration. And then lastly, let's talk about how we can use how we can overcome some energy barriers. So there are many times that we need a reaction to take place. In this case, we're going to rip apart this AB molecule and instead hook the A to the C and the B to the D. So what this is saying is that the reactants start out at this G and they end up here. So this is a spontaneous process. It has a delta G that's less than 0, but it doesn't happen right away. In fact, it doesn't happen very quickly, even though that word spontaneous is there. That refers to the G. Instead, this energy of activation barrier must be overcome for the products to get down here. Now in chemistry, you sped up reactions by heating them up, and that certainly would make these molecules go into a transition state and maybe break apart the bonds. But in our bodies, we can't handle speeding up reactions by heating them up. We also don't add chemicals in as catalysts to speed things up. Instead, we add something called an enzyme. Enzymes are proteins, and basically the enzymes help to destabilize those bonds, and we'll see some pictures about that in the next screencast, where it lowers the activation energy barrier. The delta G is unaffected. We still start out here, we end up over here. It just means it is more likely for this reaction to take place. It's easier to overcome this energy barrier. And in the next screencast, we will focus on the enzymes and how they do that.